In this video, we will begin to look at the first but unbelievably important happenings in meiosis. So if we look at the slide, we can see a lot of stuff going on here. What I want to bring to your attention, though, is I want you to notice what's happening to the overall number of or amount of genetic material in all of the daughter cells. Okay, at the end, we start off with something in meiosis and we finish with something different. And that is a really big differentiator between meiosis and mitosis. I also want to bring to your attention, which is what we're mostly going to talk about today, I want you to look at this set of slides here after, or this set of cells, after meiosis 1. Notice how each of the chromosomes no longer are one set color. They're a mixture of two colors. And this is going to be something that we're going to focus on in this video. Okay, so if we look at meiosis as an overview, we see that meiosis's job is to cut the number of chromosomes in half, whereas mitosis's job is just to make a copy. So if we remember, mitosis's job is to make a carbon copy of the DNA. So we start with 46 chromosomes and we end with 46 identical chromosomes, okay, in the daughter cells. This is not the case for meiosis. Meiosis is only used in sex cells. So that means for females, it's only used in our ovaries. And for males, it's only used in the testes when we're making gametes. Okay, so our ova, our eggs, and sperm cells. Every other cell in the body is gonna undergo mitosis. It's only the sex cells that undergo meiosis, and that's because we need to cut the number of chromosomes in half. Otherwise, an egg is going to come to the table with 46 chromosomes and a sperm cell with 46. And when you combine that, you're going to end up with 92 chromosomes. Humans don't have 92, okay? Humans have 46. So we have to cut the number of chromosomes down by half, all right? That means that with this mixing of DNA and the cutting of the chromosomes, the result in meiosis ends up being that you have 4 daughter cells, 4 potential gametes that are genetically similar, okay? Whereas in meiosis, similar, mitosis, identical. This is why your biological parents could have as many children as they wanted, and unless they have identical twins, no two offspring are going to be genetically the same. Some of you may look like your siblings, you may act like your siblings, but you are not identical to your siblings. You're very similar. So if we look at this, we see that the job, now meiosis is in 2 stages, whereas mitosis is in 1 stage only. And if we look at the overview of mitosis, we see that the names are the same as in mitosis. So we break it down as meiosis 1. Okay, so this is meiosis 1 here. And this is going to be meiosis 2. So we see that the end result here is that in meiosis 1, we go from 46 chromosomes to 23 chromosomes. And meiosis 2, the whole point here is to take those 23 chromosomes and to split them up into 23, each cell having 23 chromatids. Okay, so we go from a full complement of chromosomes to a half complement of chromosomes. And then we go from a half complement of chromosomes to a half complement of chromatids. So if we look at meiosis 1, okay, very similar to mitosis, Meiosis 1 is going to start with a cell that was in interphase, and when that cell is actually ready to basically divide and form a gamete, it's going to start to prepare itself just like any mitotic cell would, any cell going through mitosis. So we start off with prophase 1. And I want to bring to your attention here what's happening in this image. So I want you to notice that you've got two sets of similar chromosomes here. Okay, you've got your centrioles. You see that the asters are starting to form here. They're going to split up. One of these green chromosomes is going to start going this way. The other's going to go that way. They're going to form their spindles like normal. But I want you to really pay close attention to what's happening inside the nucleus. So you notice here how this orange and this yellow chromosome kind of look similar. They've got the same sort of size. They seem to also have the same sort of little bands here and there. Okay, and then the pink and the purple chromosomes are also evenly matched. When prophase 1 starts, actual chromosomes, so in your cell you have 46 chromosomes, 23 of which you got from your biological mother, and 23 of which you got from your biological father. Chromosomes 1 through 23 from the mom and 1 through 23 from the dad are actually identical in size and genetic material. So that means here that homologous chromosomes find each other. Okay? When we talk about homologous chromosomes, we talk about the fact that they are identical in size and they carry the exact same genetic code. So that means that chromosome number 1 that you got from your biological mom has the same genes on it as chromosome 1 that you got from your biological dad. Those genes might have different alleles. So one might be eye color blue and the other one might be eye color brown. But the exact gene for eye color is on the same chromosome. So these actual homologous chromosomes find each other in prophase 1. And whether they come face to face with one another or side by side with one another, they link up. Okay? And that's going to set something up called crossing over. And this is super important. All right? Crossing over actually is the reason that sexually reproducing organisms are genetically similar but not identical. So if we look at a little bit more in depth about what we mean for homologous chromosomes, we're actually talking about the fact that let's say this chromosome here you got from your mom, and this one you got from your dad, okay? They have the exact same genetic code. They're exactly the same size. So what I mean by the exact same genetic code is that, let's say in this section here, you have a gene that codes for curly hair or hair structure. Well, on this exact side, in this exact same spot on its homologous pair, you're gonna have the exact same gene. You may not have the same code. So this person here, the female, might code for curly hair, and the male might code for straight. So the male might code for straight hair. But both code for the hair texture or hair structure. Okay, so that's what I mean when I say that we've got the exact same genetic code. When these, and remember, this is all happening in prophase 1, okay? Prophase 1. When these homologous chromosomes find one another and they come either face to face, back to back, side to side, when they basically pair up, okay, that pairing up is known as synapsis. And synapsis is the first actual step of what's known as crossing over. So crossing over is actually the whole basis of sexual variation. Okay? It's because of this actual trait that sexually reproducing organisms have that make it that not one other organism is genetically identical to another unless they are maternal or identical twins. Okay? No two humans are exactly the same. All right? And that's because of what ends up happening in this image here. So if you look at this actual image, you see that there are two homologous chromosomes here. All right? The pink one has actual genetic code, and I'm going to do this in a different color, has these genes that are in these three locations. The male genetic code, or the purple one, let's say, has the same sort of deal. It's got 3 genes, but we're going to label those in a different color. So when synapsis occurs, all right, when synapsis occurs, these actual chromosomes are going to intertwine with one another. They're going to get all tangled. So what ends up happening here is you're going to see that chromosome, the first one here with its genetic material, is going to actually get tangled with the second chromosome here, and in that tangling, right down here in this tangling, this piece is going to break off and this piece is going to break off, and they're going to swap pieces. They're going to exchange DNA. So that means that the result here is that we still have, once these chromosomes are right next to each other now and they're done kind of tangling, is we still have the intact chromosome, but if we look at it now, we see that the chromosome is no longer an actual replica on both sides here. So we've got one chromosome and a second chromosome, right? And then within that, we've got one sister chromatid, a second one sister chromatid, and a second one. Once crossing over occurs, the two chromosomes are no longer the same. So that gives us 4 slightly different versions, okay, of an actual potential gamete. Now, this is super important because this is why sexual reproduction actually leads to similarity and not replication, perfect replication. So once we kind of get the gist of what crossing over is, we want to talk about why it's so important. And I've alluded to this time and time again in this video. It is super important because one of the traits of sexually reproducing organisms is that they produce genetically similar organisms or offspring, not identical. And this is why things like sicknesses and viruses and bacterial infections or disease don't wipe out the entire planet, because we're all genetically similar but not identical. And that is one of the key features of sexually reproducing organisms. Okay, so it increases the genetic variation, which is always a good thing. Okay, and it's super important to realize that whatever piece of DNA here gets swapped is completely by chance, which is actually a pretty cool thing when you think about it. Okay? Once these actual chromosomes are all set up in their homologous pairs, we refer to them, once they're different, as being recombinant. And recombinant simply means that we've recombined the genetic material. We've mixed it up again. So at the end of prophase 1, okay, so at the end of prophase 1, we have recombinant chromosomes that are ready to keep going with meiosis 1. Okay, so that means they're going to go on to metaphase and anaphase and telophase 1. And the whole point here is that we've mixed the DNA up. And we're going to get to, in the next video, what ends up happening in terms of meiosis 1 versus meiosis 2.